Okay, so we are now going to progress to part 3 of the systematics lecture. Again, remember we talked about biodiversity and how we organize all the diversity of life out there, trying to create a better picture of the relatedness between species and different taxonomic groups and so on and so forth. In the last lecture, we talked about ancestral and derived characteristics. The derived characters or features are basically what you use as those branch points, and it's shared by everything above the branch point, but not below. So back to just quickly our example here. When jaws evolved, everybody above this point had jaws. These guys didn't. That was a new feature for everything in these groups here. Lungs evolve. Everybody above lungs, above that branch point, possess those traits. So this is where, when we construct that cladogram and we get those numbers, who's got a 1, 2, 4, 6, etc., that's how we figure out where we put them. So hypothetically, if we grabbed a new species and said, I want to look at, I don't know, a chicken. Where would a chicken fit in this? And then you put the chicken right here. You go chicken, and you run through, chicken down here, and you run through the characteristics. Do they have jaws? Do they have lungs? You do all those features, you get their score, then you come here, and then you'd figure out, okay, well, where would a chicken fit? Does it fit right next to here as a 6 as well? Is it back here as a 4? Where does that particular group fit within the evolutionary picture? Okay, so we keep doing this. We keep expanding our knowledge and growing these trees. So what we're creating are different visual representations of the relatedness. So the cladogram, when we build this, the cladogram depicts a hypothesis. And I want to underline this and put it in bold to really emphasize. Hopefully you guys all remember what a hypothesis is. It's an educated guess. It's a prediction, an idea, etc. It's a hypothesis of evolutionary relatedness. So this is what we think. We're not guaranteeing it. We're not calling it a law or a theory. But this is what we think based on the evidence we have and the information we have today. Hypotheses are constantly being tested. They're being adjusted. They're being disproven. Throw it out. Didn't work. It's not right. Or continuously supported. Here's more data, more evidence, more information that supports this. So that's what a cladogram gives us, is it depicts this hypothesis of evolutionary relatedness. Now, as we build cladograms, we have things called clades that we'll start to see. Clades are, or is a group of species that share a common ancestor due to shared derived characters. Shared derived features are those traits. So everybody who's got the same whatever is going to be in a clade. So when we look at the drawings or the little diagrams here, everybody in the green box here is considered a clade, because here's the branch point, and then all of you here have this trait shared by everybody here. That makes you a common clade. We're not going to say this one also is included over here because it doesn't have this particular trait. This could be a clade, just this particular group right here, this evolutionary line. And this could be 3 different species. We'll call it A, B, C, or 1, 2, 3 that evolved down this line here. We could look at them as a clade. because they have this common feature that appeared right here that went down the evolutionary line. So something to stress when we're looking at these little diagrams, you could have multiple species along this line here. That line from there to there represents a window potentially of millions of years. That's what MY means. Millions of years. There could be half a dozen species that evolved down that line. Or we could reduce the time frame. Maybe we say this entire window is only 15,000 years. So that's a piece of information we want to include within these diagrams is the timeline, the estimated or expected time frame. Down here, this red box would not be considered a clade because you have these guys over here, and then you're trying to group this one in with them, but you have to come all the way down here to find the common trait, that common shared derived character that would be shared by all of these. And if you're just isolating right up here, that cannot be considered a clade. Same problem over here. This is not considered a clade because you're talking about a shared derived character here that is found in these two individual lines right there, but you're excluding this one. Doesn't work that way. It's kind of like saying, oh, my cousin isn't part of the family because I don't like him. You can't do that. They're part of the family. That's what a clade can be viewed as, is a family or a relatedness, a group of species or organisms that have shared derived characters. So let me highlight that just to reemphasize. To be in the clade, we look for those shared derived characters. Okay, so that becomes an important thing. Now, the big red star. Boom, boom. Big hint, when you see that, write it down. It's going to be on your quiz or on your exam. Okay? That's my way of being very non-subtle about something that you guys will be tested over. Right? I want to make sure you understand this. So I try to just be very obvious with it. Parsimony. This is an important thing in science. Principle of parsimony. Parsimony is the cladogram that is the simplest. The principle of parsimony favors a cladogram that is the simplest. Let me make this smaller so I can put a bit better of an explanation up here. The simplest explanation is the most likely. It's most likely going to be the accurate explanation. Why are these related? Because why do we see this trait in species A, B, and C? Because they have a common ancestor. The likelihood that they all evolved a trait independently, and then this showed up, and this happened, and that, not that likely. So we aim for parsimony. What is the simplest explanation? That is the most likely explanation for an event. The most accurate explanation is based on the principle of parsimony. Okay, so when we're doing cladograms, go for that. Go for parsimony. Simple is most accurate. Not a guarantee, but if I'm going to base my guess, my educated guess on it, I'm going for the simplest explanation. All right, that's general rule of life. Occam's razor, principle of parsimony, same concept. Simple is most accurate. Okay, so molecular folks in this group, those of you excited and interested in molecular research and that type of stuff, love it. DNA sequencing, awesome, phenomenal. Excited that that's been brought into cladistics and systematics and helping us with phylogenies. What we're learning is that approximately 1% difference in your DNA is equal to about 5 to 6 million years of evolutionary divergence. Okay, so if I have a cladogram that does this, and I'm looking at species A and species B, and I run a DNA sequence and I find there's about a 1% difference, I can estimate about 5 to 6 million years ago, that branch point, there was a common ancestor that gave rise to both A and B. And it took about 5 to 6 million years for them to evolve that 1% difference in their DNA. That is very general, rough estimation. But that's what those length of lines on those branch points represent, is evolutionary time. So here's an example. We have 5 species of pine trees, A, B, C, D, and then what we call the outgroup. That's just the weird one that you put in there for what they call the outgroup. And you look at a DNA sequence, and you start looking at relatedness and similarity, and you get to this, and you go, oh, okay, well, this is the evolutionary picture. A and C are closest. Then they're related to D. Then the next closest relatedness is to B, and the least relatedness is the outgroup, that bluish, green, whatever pine tree that is. So the branch length helps us figure out evolutionary time. How long is that branch? From here to here. Time. So it took so many millions of years to go back to the common ancestor right here for A and C. But consider the length of this branch. That's like 4 times the amount of time. So again, making it easy. If this is 2 million years to go to the common ancestor right here, and it would take 4 of these increments to get down here, that means this is about 8 million years ago for all of these to come back to their common ancestor, right? So that is the value of understanding the molecular and looking at changes in molecular DNA and how that gives us a rough picture of time. Now, what we can then do is let's see if we can find more evidence to support this hypothesis. Do we have fossil evidence? Do we have anatomical evidence? Do we have other lines of evidence that would support an 8 million year ago ancestor to all these species? Let's test it. And let's test it and test it and test it. And if the data supports it, we keep testing it again. If the data rejects it, we throw it out and say, okay, redesign the experiment. But this is how science is done here. This is how we look at these things and how we figure out those connections. So, okay? All right, so now when we're getting this information, we do all this work, we're exhausted, we have all this, we then move into the field of classification. So classification is where we will place species and higher groups into the taxonomic hierarchies. Oop. All right, so I hope everybody remembers domain, kingdom, phylum, class, order, family, genus, species, because I'm not going to run through that with you guys. If you don't know it, let's definitely look it up, but those are the taxonomic hierarchies. Linnaean classification. All right, and we'll talk about new classification as we get into the protists and our classification stuff in this semester, but we see that this is where we take all this information and how we put it together in a classification. So we will go into a little more detail about this monophyletic, paraphyletic grouping as we go into the next part of our lecture. So tune in, click on the next link, let's check it out.